Hello, this is Gina Piscatelli with a lecture on the female reproductive system for Anatomy and Physiology 2. And in this lecture, we're going to look at the ovaries and their function, primarily oogenesis or gametogenesis. So let's first just cover the anatomy of the ovary. The cortex of the ovary, the outer layer, is where gametes are formed. And we can see in this picture several stages of gamete formation or oocyte formation. You're not really seeing what's happening in these developing follicles, we call them, but all these different colors and round structures that are located in the cortex are different stages of oocyte and follicle development. And we'll come back to what that means in a little bit. The outer covering beyond the cortex is called the tunica albuginea, just like we had in the testis. It's a fibrous connective tissue. But the innermost region of the ovary is the medulla. And the medulla is shown here and labeled here in the middle. And this is where large blood vessels are located to supply and drain the ovary. And also there's a nerve or a few nerve fibers that come into the ovary. You know, it's pretty well established that the ovary function is controlled by hormones, but there's also quite a bit of evidence that the autonomic nervous system helps control ovarian function as well. We won't have time to talk about that in this class, but we will look at the hormonal control. So that's just some general overview of the anatomy of the ovary. Now we're going to look at the function of ovaries. One you're familiar with already because we talked about it in lab, and that's formation of a gamete, gametogenesis. That gamete also has to be released so that it can find a sperm and travel to the uterus if the gamete's fertilized. And so the release of the gamete is called ovulation. So two major functions to begin with: making a gamete and ovulating that gamete. And then a third function is to produce estrogen and progesterone. Now, estrogen and progesterone are made by certain cells in the ovary called follicle cells, and we'll come back to that too, where they're located, and there's different types. But the main purpose in producing the estrogen and the progesterone, in part, has to do with forming a gamete, but it also has to do with controlling the uterus. The uterus has a menstrual cycle. The lining of the uterus thickens to prepare for pregnancy. And if pregnancy doesn't occur, then it thins again. All of that function of the uterine lining is controlled by the ovarian hormones. So the ovary sends out hormones via the bloodstream that reach the uterus, and those hormones control the menstrual cycle. In this lecture, we won't talk about the uterus, we'll just talk about the ovary. So the ovary has these functions: gametogenesis, ovulation, production of hormones, and it performs these various functions in a cyclical pattern. We call this pattern the ovarian cycle. So when you see that phrase, what it refers to is the fact that the ovary produces gametes and hormones in a cyclical repeating pattern. The pattern repeats approximately every 28 days or once a month. The ovarian cycle will include releasing a gamete, that's called ovulation, and that occurs at the midpoint in the cycle. So approximately at day 14, and this picture shows the release of a gamete. Now the ovary is not this thing that's releasing it. This thing that's actually releasing the ovum or the oocyte is called a follicle, and we'll come back to that. So on either side of this day 14 ovulation event are two phases. Prior to ovulation is the follicular phase, and this is when a follicle grows and develops to the point where it will actually rupture and release a gamete or an ovum. It's actually an oocyte. There are a lot of things that happen during this follicular phase that we'll discuss, but it's all about developing the follicle and making sure that the oocyte is at the right stage to be released. After ovulation is the luteal phase, and it's named because a structure forms called the corpus luteum, which is really a remnant of the follicle. Once it's ruptured and released the oocyte, then it kind of involutes, so to speak, and we'll talk about what that means in a little bit. So the corpus luteum is what really defines the luteal phase, and growth of the follicle is what really defines the follicular phase. So again, the events that are going to happen during the follicular phase will be gametogenesis, but that follicle is going to make a hormone called estrogen, or many hormones that we refer to collectively as estrogens. They vary slightly in chemical composition, but you can just think of it as estrogen secretion. And the events that occur during the luteal phase is formation of that corpus luteum, and that corpus luteum is what secretes progesterone. So we have a lot of estrogen production prior to ovulation and a lot of progesterone production after ovulation. There's a little bit of variation to that, but that's a nice sort of summary about how to think about hormone secretion. Both of these hormones are since they fluctuate in quantity throughout that cycle, will control the cycle we see in the uterus. So we will focus first on how does an ovary produce a gamete, gametogenesis. It turns out that the ovary's gametogenesis involves two distinct processes, but they're dependent on one another. One is oogenesis, and that is maturation of an oocyte by the cell division process we learned called meiosis, making sure that the gamete has only a haploid number of chromosomes. The other process that takes place in gametogenesis is development of a follicle, so it's called folliculogenesis. And the follicle is a structure that's composed of several cells surrounding an oocyte. And in the diagram on the left, you can see that in the cortex, this is the site where the primordial germline cells are located that can become oocytes. And as the oocytes mature, they're shown as yellow, these oocytes. As they mature or go through the various stages, we'll see that they become surrounded by increasing numbers of cells. These are follicular cells, and collectively we call this structure that surrounds that yellow oocyte a follicle, made up of many different cells, but nonetheless we call it one thing a follicle. So this follicle will grow to a certain size and release the oocyte. And as I mentioned previously, after ovulation, what forms is this structure called the corpus luteum. The corpus luteum eventually can degenerate, especially when pregnancy doesn't occur, and you get this formation of another structure called a corpus albicans. And I'll mention that again later. But all of this is happening in the cortex. All right, let's talk about follicles first, because they really are the functional units of gametogenesis, which is our topic to begin with. These follicles, I've got many of them circled in this picture, histological picture of an ovary. And at the very periphery, you can see that they're super small. So my original circle has many follicles circled. But then once mature, they become quite large, as you can see in this one right here. What they do is surround the oocyte and provide an ideal environment for the oocyte to mature. Now, not all follicles will go through this process during a woman's life. Some are recruited to grow and release a mature oocyte, and that's dependent on hormone sensitivity. And we'll talk about how many actually are recruited to grow to the large size. So the variation in the size and structure of these different types of follicles, because they obviously look different, and there's lots of them, reflects different stages of their growth, the follicle's growth. So we're going to talk about how follicles change over time and why that's important for oocyte development. So the stages of folliculogenesis include, first, we start off with a primordial follicle, and that follicle is surrounding again a yellow oocyte with a blue nucleus. And this primordial follicle has one layer of squamous cells. So you could say surrounded by simple squamous, and it is an epithelium. But over time, that primordial follicle changes, and we'll talk about how in just a second, to a primary follicle. Instead of a primordial, now it's a primary follicle. And this occurs when those squamous cells become cuboidal. After that, the cuboidal cells multiply by mitosis, and so you get layers of cells. Now we have a stratified cuboidal epithelium, and we could call this a secondary follicle just because it has more than one layer of cells surrounding the oocyte. But in addition, those layers of cells differentiate amongst themselves. So the outer cells become what are called the thecal cells, and the innermost cells become granulosa cells. So this secondary follicle has an oocyte in the middle with granulosa cells surrounding it, usually 4 to 7 layers, and then a layer of thecal cells, maybe 2, surrounding those. In addition, we see this clear structure. It's a fluid-filled space between the oocyte and the granulosa cells. That structure is called the zona pellucida. So you can name a secondary follicle by the fact that there's layers of cells and that you see a very thin, clear zona pellucida around the oocyte. The fourth stage of folliculogenesis occurs when the granulosa cells have secreted so much fluid that the fluid forms this cavity in the center of the follicle. That cavity is called an antrum. We still have thecal cells and we still have granulosa cells, but notice the oocyte isn't in contact with all these granulosa cells. In fact, the oocyte is connected to the follicle by just this simple stalk here, and we'll talk about that again. The fifth stage of folliculogenesis is formation of a tertiary follicle, also called an antral follicle, but really you could call either one of these. It's rupturing. It's rupturing what it is the fifth stage, is rupturing of this tertiary follicle. And that occurs because so much fluid has been secreted by the granulosa cells that pressure has increased and caused the follicle wall to burst. And when it bursts, it releases the oocyte and its surrounding granulosa cells. So you'll see that there's granulosa cells still surrounding this oocyte. So it's still protected. And then the next stage would be the follicle turning into a corpus luteum, which is a yellow structure. And then if pregnancy doesn't take place, implantation of the embryo, that corpus luteum degenerates into kind of fibrous scar-like tissue called the corpus albicans. So the follicles at these different stages of development are distinguished from each other by their size and structure. The first follicles to develop in the fetus are called primordial follicles, and there's approximately 7 million primordial follicles, each containing an oocyte, a developing oocyte in the fetus. During fetal development, only 2 million survive all the way until the time of birth. So the other 5 million or so have undergone a process called atresia, which is a process of programmed cell death. They essentially degenerate and they're not maintained in the ovary. But the remaining 2 million survive at the cortex of the ovary. Until the onset of puberty, most of these primordial follicles don't change, they don't develop. A few more undergo atresia, so not even all 2 million will survive from birth to puberty. Those that do survive, many will just stay at this primordial follicle stage. It's estimated there's about 400,000 left at puberty. Most of them will stay because if you think about it, each one of the 400,000 contain an oocyte, and a woman only releases one oocyte every month. So that's 12 a year. And if she has a reproductive life of 40 years, that's just over 500 or so oocytes that she'll use. And there's 400,000. Clearly they won't all progress beyond this primordial follicle stage. So when you hear people referring to the fact that a woman is born with a lifetime supply of oocytes, what they're referring to is the fact that there are approximately 400,000 follicles, primordial follicles, each one containing an oocyte or at least an oogonium. Okay, it might not have matured yet beyond that stage, but each one containing a potential gamete. From puberty until a woman reaches menopause, a group of about 20 primordial follicles are recruited, they say, each month to continue this process of folliculogenesis. What this means is that each month, 20 or so primordial follicles will respond to changes in hormone levels. These are gonadotropins coming from the pituitary gland. But then even those 20 that have responded to gonadotropins from the pituitary gland won't make it all the way through this process that you see here, these stages of folliculogenesis. In fact, just one is going to become what is known as the dominant follicle, and it will then progress through all these stages. It takes about 1 year for a single recruited primordial follicle to progress to the ovulation stage right here, rupture of that tertiary follicle. Let's look a little bit more closely at particularly a secondary and tertiary follicle so that you understand the difference between the cells that exist inside that follicle. So I'll just start with recognizing first a primordial follicle because we were at the beginning and it makes sense. So here's the first 3 stages of folliculogenesis. We have this primordial follicle. It exists in the cortex, the most superficial layer of the cortex. And around the oocyte is one layer of squamous cells. And I've got it circled on this histology picture on the left. So it's yellow, circled in yellow. The oocyte's in the middle, and the squamous cells surround that oocyte. The next stage is development of a primary follicle. Now the oocyte's a little bit bigger. That has to do with gonadotropin hormones as well. And the layer of cells have changed. The cells have changed in that one layer. They're now cuboidal. Sometimes you can see a zona pellucida already, but usually not. And I have that circled in red in the histology picture on the left. So that's an example of a primordial follicle. And I distinguished it because these cells are starting to look a little cuboidal. The next stage is that the primary follicle, through the process of mitosis, will form multiple layers of cells. And now it's going to be called a secondary follicle. And again, the oocyte becomes larger in the center. So that picture on the left shows a secondary follicle circled in green. The oocyte is in the middle, and then you see at least 2 layers, but really more because I can see these outer layers here too, of cells, multiple layers of follicle cells. The innermost ones are called granulosa cells, and they're cuboidal shaped. So I see 2 layers here of cuboidal shaped cells. And then on the outer perimeter of this follicle are more flattened cells. They're called thecal cells. And you can clearly see the zona pellucida. It's filled with fluid, and that fluid was made by the pink granulosa cells, the cuboidal cells. So we're accumulating fluid around the oocyte. Now, I like talking about secondary follicles because it sets up what you have to know in the future about ovary function and its effect on the uterus. The outermost cells, called thecal cells, make androgens, such as testosterone. The granulosa cells make estrogens. And in addition, they make an enzyme that's able to convert testosterone or any other androgen into estrogen, keeping estrogen levels high around that oocyte. And that's going to help promote the oocyte enlargement. In addition, the zona pellucida contains these glycoproteins that were made by granulosa cells. So the zona pellucida has this fluid with glycoproteins. And those glycoproteins, they have certain names, ZP1, ZP2, blah, blah, blah. They facilitate sperm binding. They prevent polyspermy, which means too many sperm having access to the egg. You don't want more than one. And a lot more. They really protect interspecies fertilization. They protect against a lot of things. So the zona pellucida, it looks like it's just a nice cushioning for the oocyte, but in fact there are chemicals in there called glycoproteins that are going to help with fertilization. Okay, now let's progress on to the tertiary follicle. The tertiary follicle has 6 to 7 layers of granulosa cells, plus then layers of thecal cells on the outside. So the reason that this tertiary follicle is called tertiary or antral is because it's denoted or designated as such because of the presence of an antrum. So this fluid-filled cavity that's just huge is called an antrum. Again, that fluid's been secreted by the granulosa cells, and notice that it's separate. It's a separate space. The antrum is a separate space from the zona pellucida because the zona pellucida is right next to the oocyte. Instead, the antrum is separated, the fluid in the antrum is separated from the oocyte by a ring of granulosa cells that have been termed the corona radiata. And the whole corona radiata, suspending this oocyte, is attached to the rest of the follicle wall by this structure called the cumulus oophorus. So the cumulus oophorus creates a chemical and physical barrier between the blood and the oocyte. The corona radiata does as well. And so what that means is that anything that's going to get to the oocyte has to go through the cumulus oophorus. The thecal cells that are surrounding the outside of the follicle are of two types. The outermost layer is called the thecal externa. They can contract when it's time to increase pressure in this antrum, and that will eventually facilitate rupturing and facilitate release of the oocyte or ovulation. The theca interna cells, they look a little bit pinker. Looks like there's two layers, and they're more squamous than the small cuboidal cells that are granulosa cells. But this region, the theca interna cells, those are the ones that make androgens, like testosterone. Now, in the next stage of folliculogenesis, the antral follicle is going to rupture to cause ovulation and formation of the corpus luteum. So during this stage, the cumulus oophorus, or the stalk, constricts, and that causes the oocyte and corona radiata to float freely. But those thecal cells that were on the outside, the external thecal cells, they can also kind of constrict and put pressure on this antrum, and that causes the follicle to rupture. What is released, and this is what's most important, what is released from an antral follicle is an oocyte surrounded by the corona radiata granulosa cells. Once these structures leave and the fluid leaves, then the follicle kind of collapses. And since it had a blood supply specifically through that cumulus oophorus, there's going to be some bleeding. There's going to be some bleeding into the antrum. Eventually the blood clots and it's absorbed. Now the granulosa cells that remain and the thecal cells not drawn on this picture on the outside are going to multiply by mitosis and fill up that antrum and of course repair the wall, so to speak. And so what you get is this thick structure called a corpus luteum. Now lute means yellow, and yellow, the yellow color that's always, you can actually see it in an ovary, it looks kind of yellow, is due to the accumulation of lipid in theca interna cells. So they're just part of the follicle cells that accumulate this yellow lipid. They're called lutein cells. The whole structure is called a corpus luteum. The reason we stress the formation of the corpus luteum, even though we've lost the oocyte, so to speak, it's not even in the follicle anymore, is because the corpus luteum secretes progesterone. And I bet you've heard of progesterone before. It's another one of the ovarian hormones, right? And we talked about it at the beginning of this lecture as well. Okay, I have a couple of videos for you to watch. I'm going to see if this works. It may or may not work. We're going to try it. This is ovulation, a drawing format from the University of New South Wales in Australia. So we have formation of the secondary, tertiary, antral follicle, ovulation. That's kind of cool. It's a drawing though. This one's even better. And I hope the lighting's good enough for you. This is an actual ovary. It's a rabbit. But you can see release of the oocyte from the surface of the ovary. And you can even see another, I think I can draw on it, another site on the surface of the ovary where maybe next month ovulation could occur. Okay, let's talk about the corpus albicans. The corpus albicans is the last stage of folliculogenesis, formation of the follicle. Granted, I don't know if we can really call either the corpus luteum or the corpus albicans a follicle anymore. They're not either one holding an oocyte, but they're part of their stages of what happens to the follicle cells, so we put it with folliculogenesis. So the corpus luteum is maintained if pregnancy occurs, and that's a hormonally regulated thing that we'll talk about later. But if pregnancy does not occur, then what happens is the corpus luteum degenerates, the cells die, they're not going to make progesterone anymore, and you get this scar-like formation called a corpus albicans. And up in this picture, you can see that dotted white line. Somebody has circled at least part of a corpus luteum, and this was like the center of it right there. I'm sorry I didn't get it all. But in the very, down a little bit lower, you can see where there used to be a corpus luteum that has now turned into a corpus albicans. Okay, let's just summarize what we looked at with folliculogenesis. Basically, I was saying that the follicle is the functional unit of the ovary because it's housing the oocyte, and it's the follicle that becomes dominant within a 1 month cycle. And so whichever follicle develops the best and is furthest along is the one that will release the oocyte. So folliculogenesis is really important to releasing a gamete. Primordial follicles in an adult woman make up about 90 to 95% of all of her follicles. And you can see at the periphery of this ovary how numerous those primordial follicles are. Each month, a group of those are recruited by a hormone called follicle stimulating hormone, FSH, to become primary follicles. Within the primary follicles, cells divide by mitosis to form a secondary follicle. So there's layers of cells. So I'm going to pretend that this is just one layer of cells surrounding an oocyte. I think it is. That would be a primary follicle. Whereas the one above it would be a secondary follicle because there's layers of cells. And when there's layers of cells, we get two types forming. The granulosa cells that make the zona pellucida and secrete estrogen, and the thecal cells that secrete androgens. Now those androgens can be turned into estrogen by the granulosa cells as well. Eventually what happens is this secondary follicle becomes either an antral follicle or a tertiary follicle. And that those are noteworthy or distinguished by the fact that you have this fluid-filled cavity in the center of the follicle. And that's called an antrum. So the granulosa cells are the ones that made the fluid that aggregates together, pushing the cells out of the way and forming this cavity called the antrum. Eventually, the tertiary or antral follicle ruptures and an oocyte is ovulated, and that oocyte is surrounded by the corona radiata. Eventually, the follicle collapses, forming a corpus luteum that fills with blood and that yellow lipid. But the cells are alive and they secrete progesterone, which is going to affect the uterus. The corpus luteum eventually degenerates, we say involutes, shrinks, whatever, to scar tissue, and then it becomes a corpus albicans. So that's what's happening with all the follicles in the ovary. Half of gametogenesis is the process of folliculogenesis, changing these follicle stages. But what about the oocyte? What stage is it in when it's ovulated? So let's look next at the stages of oogenesis. This is when we talk about the development of the oocyte. And it turns out that the stages of oogenesis are somewhat independent from the stages of folliculogenesis, at least in terms of stage numbers like primary, secondary, tertiary. And I say that because although the primordial follicles most likely have a primary oocyte, we also see a secondary follicle containing what's known as a primary oocyte, or maybe a primary follicle versus a primordial follicle. So the primary oocyte stage exists in three types of follicle stages. So in a primordial follicle, the oocyte could still be primary. It is, I'm sure. And then in the primary follicle stage, it's still a primary oocyte. So the follicle's changing without the oocyte changing, okay? When we get to this tertiary follicle stage, the antrum is present, and now we never get to a tertiary oocyte. That doesn't exist. This could either be a primary or a secondary oocyte. So it's a little bit independent. So we're going to discuss just oogenesis by itself and then look at the two processes side by side for naming conventions. So as an overview, the embryo has germline cells that more specifically are called oogonia in the ovary. These are the ones that are destined to they're stem cells, and they're destined to transform into a primary oocyte that would undergo meiosis. So during fetal development, as the fetus gets bigger and bigger, these oogonia multiply by mitosis, just like the rest of the fetus does. Some of them are going to transform into primary oocytes, and it turns out by birth, all of the oogonia have been transformed into primary oocytes. During infancy and childhood, these primary oocytes begin meiosis 1, but they stop right away in late prophase 1. Now you might remember that it's prophase 1 where the homologous chromosomes line up and cross over. So if I draw my centromeres in here, this picture might make more sense to you. So I'm going to put a centromere there and a centromere there. Oops. And what we have are two homologous chromatids crossing over and exchanging information. So synapsis has happened. So this is where everything is arrested in prophase 1 until puberty. Then at puberty throughout adulthood each month, a primary oocyte can complete meiosis 1. Now it's going to depend on what follicle it's in. If the follicle has been recruited, then it will do so. So at puberty, a single primary oocyte will complete meiosis 1. I don't mean that just one does, a group does each month. And eventually we will get a secondary oocyte, but it's going to depend on which follicle becomes dominant. Which one goes all the way to the secondary oocyte stage, okay? So the secondary oocyte that forms each month begins meiosis 2, and it stalls in metaphase 2. So what you have in this case is an oocyte. I better draw a picture. An oocyte with chromosomes in metaphase 2, so already haploid, and the chromosomes are lined up individually at the metaphase plate, as if it were mitosis. I'm trying to draw in a centromere. It's not working too good. This one already has one. Okay, so this is the stage of an oocyte that's ovulated. A secondary oocyte is in metaphase 2, which automatically, since it says 2, is meiosis 2. If fertilization does occur, meaning if a sperm is around and touches the surface of this secondary oocyte, it will complete meiosis 2 and produce a mature ovum that exists just briefly until the sperm nucleus joins with the secondary oocyte nucleus to form a zygote. That was a lot of information, so let's relate it to what we learned in lab. Just review oogenesis and compare what I just said with what we learned in lab. So we talked about the fact that these are germline cells set aside in the ovary to potentially undergo meiosis. These are really called oogonia. That's plural. Just one is an oogonium. And they multiply, but then they differentiate into primary oocytes before birth. These are still diploid cells. Then they begin each month. Well, the primary oocytes start meiosis 1 before birth. But they're arrested in meiosis 1, prophase 1. Then each month, from puberty until menopause, a group of these primary oocytes are selected in the follicles that they're in to undergo further development. And at that point, we can get further progression. Meiosis 1 will complete. Meiosis 2 will begin. But meiosis 2 will be arrested at the metaphase 2 point. And in fact, meiosis 2 will not be completed unless a sperm comes to penetrate the egg. So if that happens, then you get division of both a polar body and this secondary oocyte. And the products will be more polar bodies and an ovum, all of them haploid, containing half of the genetic information. One more time, just to make sure it sticks a little bit. We start with oogonia in the ovary, and they all transform into primary oocytes before birth. Here they are. They begin meiosis 1 before birth. At birth, all of these primary oocytes, there's like 2 million of them or something, are arrested in prophase 1. They just sort of stop. Then, after puberty onset, all the way until menopause, each month a group of these primary oocytes are recruited to start changing, to start the process. Mainly it's the follicles that are going to start changing. Whichever follicle becomes dominant, and there's just going to be one a month, that's the one follicle and the one oocyte inside of it that will complete meiosis 1 to form a secondary oocyte and a polar body. As you know, it's this secondary oocyte that is ovulated, but it has already begun meiosis 2 before the sperm came along. The secondary oocyte is actually arrested in metaphase 2. Then when the sperm comes along, now we actually get meiosis 2 completing. And so you'll get one ovum and another polar body, but the polar body that already existed will divide into 2. So the bottom line is you get 3 polar bodies and one functional ovum that contains the sperm nucleus. So that's a little bit of lab review in addition to kind of incorporating the follicle. But I think it's kind of nice to go through what happens now simultaneously with the oocyte in oogenesis and with the follicle in folliculogenesis. So this is a primordial follicle that contains a primary oocyte that's been arrested in prophase 1. Now notice that this primordial follicle changes into a primary follicle, but nothing's happened to the primary oocyte, of course, because this can happen before birth, you know, it can happen anyway. So these primary follicles typically still contain primary oocytes. Even secondary follicles still contain primary oocytes. It's a bad way to name things, really. But anyway. And then the follicle changes to an antral follicle. Well, at that point, we have to start thinking about the fact that we're close to ovulation, right? So what that means is that puberty onset's already occurred. There's been some hormonal changes. And the secondary oocyte has started meiosis II in response to those hormonal changes each month. And so now we have a secondary oocyte, but it's not done with metaphase 2 because you know you need a sperm to complete it. So it's this secondary oocyte that's present in the antral follicle. Eventually the ruptured follicle releases a secondary oocyte, and if a sperm comes in, then we'll finally get a true ovum and eventually a zygote because of fertilization or union of the nuclei. Once the follicle ruptures, now it becomes, all the rest of the cells form this corpus luteum. Kind of complicated. So let's just compare what we know between oogenesis and spermatogenesis. We know that the products of meiosis are a little bit different between males and females. We know that meiosis, forming a mature gamete, is a little bit different as far as how well it progresses or if there's any periods of interruption. We also know that the release of gametes occurs slightly differently. So let's first just look at the products of meiosis. In males, meiosis produces 4 functional spermatids. They're not completely mature yet. They need to mature in the epididymis, but they're all functional. They're not going to degenerate or anything. In females, meiosis produces 4 end products as well, but only one is functional, the ovum. Concerning the progression of the stages of meiosis, males, there's no period of interruption at all. Each stage continues sequentially, one after the other. In females, meiosis is halted at 2 points. First, in prophase 1, and that happens from, the point of embryonic development all the way up until puberty and ovulation, or I should say puberty, okay? And then each month there's the potential that at least a group of these oocytes will undergo meiosis II, but then we have an arrest again. The arrest is in metaphase 2, and metaphase 2 won't continue until fertilization occurs. The third difference is how the gametes are released. There's some similarities and some differences. So in males, spermatids are continuously released all day long, every day, due to the contraction of those Sertoli cells that surround the forming spermatids. And then eventually what causes the developing spermatids to be released from the seminiferous tubules was just the movement of fluid. In females, there's a release of just one gamete, secondary oocyte, each month. Not a bunch every day. And its release is dependent upon growth of the follicle and rupture of the follicle. Again, part of this is due to fluid, though, because there's fluid development in that antrum, and the pressure will build, causing the rupture. So that's a little bit similar, but it's not a flow through a tube, a flow of fluid through the tube. Okay, at the very beginning, We're almost done. Stick with me. At the very beginning of this lecture, I mentioned the fact that there's an ovarian cycle of function. So let's just review the phases, if you remember. The ovary is seen to exhibit two distinct phases as it functions throughout the month. The first is called the follicular phase, and that is the function of the ovary prior to ovulation. You can see now why it's called the follicular phase. It's because the follicles, whichever ones have been recruited, are going to develop. They're going to change. More cells are produced. An antrum is formed. Then ovulation occurs. The second phase is called the luteal phase. And in the luteal phase, once ovulation has already taken place, there's no need for these follicle cells to do the same thing anymore. And instead, what happens is they form the corpus luteum. And the corpus luteum might degenerate into the corpus albicans, or it might persist if pregnancy occurs. Now, of course, the ovary still has, this doesn't just happen once in your lifetime, so the ovary still has tons of follicles and oocytes within those follicles that can be recruited the next month as well. So these ovarian phases, the follicular phase and the luteal phase, are also noteworthy for which hormones are produced when. So we know that the granulosa cells produce estrogen. Well, the granulosa cells are most numerous prior to ovulation. And we indeed do see an increase just prior to ovulation of estrogen production. We see a huge spike due to the fact that there's this dominant follicle that becomes really big and makes the most estrogen. It's like a super producer, which is why it's dominant. And then in the luteal phase, we'll see that the hormone that's most prevalent is progesterone. And that's because cells of the corpus luteum produce progesterone and some estrogen. But the one we think about the most is the progesterone, because this progesterone changes the uterine lining to help maintain a pregnancy or an adequate uterine lining. So the next topic will be the uterus, and we will look at what the functions of estrogen and progesterone are. Estrogens have a couple of functions supporting the growth of the follicle and development of the oocyte. But the function on the uterus is to cause thickening of the endometrium in preparation for embryo implantation. So the initial thickening that has to happen is a result of estrogen. Progesterone maintains this endometrium as well as a placenta during pregnancy. It increases the blood supply to the endometrium through vasodilation. It causes the endometrium to secrete glycogen-rich fluid that helps nourish the developing embryo. But another function is to suppress uterine contractions during pregnancy so that premature birth doesn't occur. Thank you for listening to all of that. I appreciate it.